Welcome back to the deep dive. All right, today our mission is pretty laser focused.
Very focused.
We're launching a critical deep dive into, well, the dual language of cellular communication. We know you've got two huge chapters on your plate, electrical signaling and neurons, and then chemical messaging for, everything else.
And we know you're likely prepping for a test.
Exactly. So we're cutting through all the noise. We're here to give you the essential vocabulary, the core mechanisms, and the connections you absolutely have to master.
That's right. I mean, think of this as building cellular fluency. We're exploring two really fundamental systems.
Wow.
On one hand, you have the nervous system, this fast, precise electrochemical language that operates in milliseconds.
Super fast.
And then on the other, you have this slower, broader, and incredibly complex chemical language that coordinates everything else. Metabolism, growth, immune response, you name it.
Yeah. So our goal today is sharp. We want to equip you with more than just definitions. We want to give you context. We'll untangle the Hodgkin cycles, make sense of something like the Goldman equation, and map out the RTK and IP3 cascades.
So you can walk into that test feeling prepared and, more importantly, really understanding the molecular logic of life.
Let's do it. Let's start with the nervous system's architecture, the physical and electrical blueprint.
Okay, so when we talk about the layout, it's pretty straightforward. There are two main divisions. You have the central nervous system, the CNS.
Brain and spinal cord.
Exactly. And then you have the peripheral nervous system, or PNS, which is basically all the nerves carrying information to and from the CNS.
And inside these structures, we find the main communicators, the neurons, which are the ones transmitting the impulses. But I'm looking here and it says the glial cells are actually the most numerous cell type, especially in the CNS.
They are, by a lot.
So if the neurons are doing all the work, why so much support staff?
Because the neurons are, well, they're delicate. And they are incredibly metabolically demanding. Glial cells are their life support system. For your test, you really need to remember 4 key types.
Okay.
First, the microglia. Think of them as the immune cells, the scavengers. They clean up debris, fight off infection. Then you have the insulator.
The ones that make the myelin sheath.
That's them.
Yeah.
They have oligodendrites in the CNS and Schwann cells out in PNS. They both wrap themselves around axons to form that protective, high-speed myelin sheath.
And the last one's the gatekeepers.
Those would be the astrocytes. And they are vital. They form this physical and chemical barrier that tightly, and I mean tightly, regulates what gets from the blood into the brain tissue.
Yeah.
To create the blood-brain barrier.
Essential stuff. Okay, so zooming in on the neuron itself, it has these two major processes. Signals come in through the branching parts, the dendrites.
Right.
And then after processing, the signal gets sent down that long extension, the axon.
And we have to be really precise with the axon's parts. The cytosol inside, the internal fluid, is called the axoplasm. The myelin sheath isn't continuous. It has these tiny little gaps.
It's nodes of Ranvier.
The nodes of Ranvier. And they're critically important for signal speed, which we'll get to in a moment.
Right.
And all of this communication, whether it's electrical or chemical, it happens at the junction, the synapse. That's where the nerve cell meets its target.
Okay, let's get to the electrical foundation. Every living cell has a membrane potential, the VEEM. It's the voltage difference across the membrane. And in resting neuron, we call this the resting membrane potential. The classic example from the squid giant axon is around, minus 60 millivolts?
That's the one. And that negative charge is constantly being generated and defended. It's mainly caused by positive ions flowing out, mostly potassium.
And that's driven by its concentration gradient.
Exactly. The potassium flows out through what are called leak channels, which, and this is key, are always open. So as more positive ions leave than enter, they leave behind all these trapped negatively charged anions inside the cell. That's what establishes the voltage difference.
But wait, if those leak channels are always open, wouldn't the system just eventually, run down? Wouldn't the concentration gradients collapse?
An excellent question. And that's where the NAL plus K plus pump comes in. You absolutely have to understand its role and its cost.
It's an active system, right? Uses ATP.
A ton of it. It's an active transport system that is constantly working against those leaks to maintain the ion imbalance. It pumps 3 sodium ions out for every 2 potassium ions it brings in.
And the cost.
This one process is so critical, so non-stop, that the NAM plus K plus pump is responsible for nearly 40% of your entire body's basal metabolic rate.
Wow. That is a staggering cost just to hold a negative charge.
But it's all to set the stage for the main event, the action potential, that incredibly rapid, all-or-nothing flip in potential, from negative to positive and back again, all in a few milliseconds.
To really get the action potential, you have to look at the math, right? First, there's the Nernst equation.
Right, and Nernst tells you the theoretical equilibrium potential for a single ion. But a real cell is permeable to more than one ion, even if it's just a little bit. So Nernst isn't enough to calculate the real resting potential.
So if a test asks for the most accurate way to calculate it, the answer has to be the Goldman equation.
Precisely. The Goldman equation is the complete story. It accounts for the combined effects and, crucially, the relative permeabilities, the pK, pNa, PCl of all the relevant ions.
That what works.
It works perfectly. If you plug in the known values for the squid axon, Goldman gives you minus 60.3 millivolts. It's a dead-on match for what's measured.
Okay, so let's walk through the firing sequence. It's all mediated by voltage-gated channels, which respond to changes in that membrane potential. So when a stimulus gets the membrane to the threshold potential, the depolarization phase kicks off.
And this starts that classic positive feedback loop, the Hodgkin cycle. Once the voltage-gated sodium channels open, sodium rushes in. That influx causes more depolarization, which opens more sodium channels. It's this exponential runaway process that drives the potential way up to around plus 40 millivolts.
But that spike has to be shut down just as fast. That's the repolarizing phase.
Correct. Two things happen almost at the same time. The sodium channels rapidly inactivate, they slam shut, and the voltage-gated potassium channels, which are a bit slower, finally open up.
Which lets potassium rush out.
Exactly. Driving this potential back down towards the negative resting state. And this is often followed by a brief hyperpolarizing phase or an undershoot. The potential actually dips below the resting potential for a moment.
Why does it do that?
It's because those potassium channels are slow to close. So for a brief moment, the membrane is even more permeable to potassium than it is at rest.
Now, the cell has to control its firing rate. I see two mechanisms here. First, the absolute refractory period.
And that's absolute for a reason. During this time, the sodium channels are physically inactivated. It's chemically impossible to trigger another action potential, no matter how strong the stimulus is.
And after that?
You have the relative refractory period. Here, a second action potential is possible, but it's much harder to trigger. That's because the cell is still hyperpolarized from that undershoot, so you need a much stronger stimulus to get back to the threshold.
Speed is everything for the nervous system. A simple passive spread of charge would just fade out over distance.
It would die out very quickly.
So the action potential has to be actively propagated along the membrane to keep its strength.
And this is where that myelin sheath really pays off. Instead of a slow, continuous propagation, the signal literally jumps between those tiny exposed gaps in the myelin.
The nodes of Ranvier again.
The very same. This is called saltatory propagation, and it massively speeds up transmission. The action potential only has to be regenerated at those nodes, which saves a ton of time and energy.
Okay, we've reached the finish line, the synapse. The electrical signal now has to cross the gap to the next cell. First, you have the super fast electrical synapses.
Right, which are basically just gap junctions. Ions flow directly, so there's almost no delay.
But most are chemical synapses. They're separated by that fluid-filled synaptic cleft.
And here, the electrical signal has to be converted into a chemical one, the neurotransmitter.
And that whole conversion process is all about calcium. When the action potential arrives, it opens voltage-gated calcium channels, and you get this rapid influx of calcium ions.
That influx is the trigger. The calcium stimulates the fusion of these neurosecretory vesicles with the plasma membrane. It's an incredible process orchestrated by these docking proteins called snares.
T snares and V snares.
Exactly. Think of them as a molecular zipper. It's a very precisely timed mechanism. For really high-speed transmission, some vesicles even do what's called kiss-and-run exocytosis. They just quickly release their cargo and then pull back.
So once the neurotransmitter is in the cleft, it binds to receptors. And these can be either excitatory, causing depolarization, or inhibitory, causing hyperpolarization.
And functionally, we can split those into fast and slow receptors. The ionotropic receptors are the fast ones. They're just ligand-gated ion channels that open instantly.
Like the nicotinic acetylcholine receptor.
Perfect example. It binds acetylcholine and immediately opens a sodium channel, excitatory. In contrast, the metatropic receptors are slower. They work indirectly through other messengers. A classic inhibitory example would be GABA, which binds a receptor that opens a chloride channel, hyperpolarizing the cell.
And a neuron almost never fires because of just one single input. It's an integrator. It's summing up all these postsynaptic potentials, or PSPs.
Right, and they sum in two ways. You have temporal summation, where one input fires over and over again really fast. And you have spatial summation, which is when multiple inputs arrive at the same time from different places.
And that signal has to be shut off quickly. I see two main ways.
Two main ways. First is just enzymatic breakdown, like a acetylcholinesterase chewing up acetylcholine. The second is neurotransmitter reuptake. This uses special transporters to pump the neurotransmitter back into the presynaptic cell.
Which is clinically really important.
Hugely. A lot of modern antidepressants, like Prozac, work by blocking the reuptake of neurotransmitters like serotonin. This keeps more of it in the synapse for longer, boosting its effect.
So that electrical speed is amazing for movement and sensation, but it's mostly one-to-one. When we step outside the nervous system, the body needs signals that are, well, slower, broader, more complex.
And that's where the language of chemical signaling really comes into its own. It's all about distance. We classify these signals based on how far the messenger travels.
The long-range ones are the endocrine signals.
Yep, they travel through the bloodstream. These are your classic hormones, insulin, cortisol, things like that.
Then you've got the local signals. Paracrine signals just diffuse to nearby cells.
Right, and autocrine signals act back on the very same cell that produced them. And finally, juxtacrine signals actually require physical contact between the two cells.
But in every one of these cases, the messenger molecule is the ligand, and the protein it binds to is the receptor. The whole process from binding to the final change in the cell is called signal transduction. A key concept here seems to be receptor affinity, and it's defined by the dissociation constant, the KD.
Right, and the KD can be a little counterintuitive. It's the concentration of free ligand you need to occupy half of the receptors. So a low KD actually means the ligand binds really tightly. It means you have high affinity.
I see. So low KD, high affinity.
Exactly. And that high affinity allows for this incredible phenomenon of signal amplification. This is where one single ligand molecule binding to one receptor can trigger a massive cascade inside the cell. We're talking one molecule of epinephrine causing the release of hundreds of millions of glucose molecules. It's stunning efficiency.
And for pharmacology, you have to know agonists and antagonists.
You do. Agonists bind and activate the receptor, mimicking the natural signal. Antagonists bind the receptor, but they just sit there. They block the natural ligand from getting in and activating it.
And cells can't just be on all the time. They protect themselves through receptor desensitization.
Yeah, they adapt. After being stimulated for too long, they'll temporarily stop responding, often by pulling the receptors inside the cell or modifying them.
All right, let's get into the workhorses of all this. The G protein-coupled receptors, or GPCRs, their signature feature is that they cross the membrane 7 times.
7 transmembrane alpha helices, that's the classic GPCR structure. And a GPCR is useless without its partner, the G protein itself.
Which is a molecular switch.
The perfect molecular switch. It's a heterotrimer G-alpha, G-beta, G-gamma, and it's active or on when it's bound to GDP, and inactive or off when it's bound to GDP. The G-alpha subunit can slowly turn itself off by hydrolyzing that GTP back to GDP.
But what if the cell needs to shut the signal down even faster than that?
Then it uses RGS proteins regulators of G protein signaling. These are basically GAPs, or GDPase activating proteins. They dramatically speed up that GTP hydrolysis, acting like a timer to make sure the signal is brief and precise.
Okay, one of the most famous pathways here is the CAMP pathway. The second messenger is cyclic AMP.
Synthesized from ATP by the enzyme adenyl cyclase. And CAMP's main target inside the cell is protein kinase A, or PKA. PKA then goes on to phosphorylate all sorts of other proteins, causing things like glycogen breakdown.
And this pathway has some pretty terrifying clinical connections.
Oh, absolutely. Cholera toxin, for instance, it locks the G-alpha subunit in its active GTP-bound state. It can't turn itself off. So you get chronic runaway activation of adenyl cyclase, massive KMP production, and that's what causes the life-threatening diarrhea in cholera.
Wow, that definitely makes the mechanism memorable. Okay, what about the other big pathway, the IP3-DAG calcium 1?
Right, so here the enzyme phospholipase C cleaves a membrane lipid, the IP2, to generate 2 different second messengers, inositol 1145 trisphosphate, or IP3, and diacylglycerol, or DAG.
And they do different things.
Completely different things. IP3 is water-soluble, so it diffuses into the cytosol, finds the endoplasmic reticulum, and opens a ligand-gated calcium channel, releasing a burst of calcium into the cell.
DAG.
DAG stays in the membrane. It works together with that released calcium to activate another enzyme, protein kinase C, or PKC. And you can't overstate the importance of calcium itself as a second messenger. It often works by binding to a protein called calmodulin, which then regulates tons of other cellular processes.
Okay, last major category. The enzyme-coupled receptors. Let's focus on the receptor tyrosine kinases, the RTKs.
These are receptors for things like insulin and other growth factors. They're unique because the receptor itself is an enzyme that phosphorylates tyrosine residues when it's activated.
And the activation starts with the ligand binding, which forces the receptors to pair up to dimerize.
Exactly. And that dimerization triggers autophosphorylation, where the two receptors in the pair phosphorylate each other on multiple tyrosine sites.
So what's the point of creating all those newly phosphorylated tyrosines? What do they do?
They become high-specificity recruitment centers. They attract other proteins from the cytosol that have a special binding module called an SH2 domain. Think of the SH2 domain as a plug that only fits into a phosphorylated tyrosine socket.
And this recruitment often kicks off the most famous cascade of all, the RAS pathway. RAS is another G protein, but it's a small monomeric one.
Right, and unlike the trimeric G proteins, it needs help to turn on. It needs a GEF, a guanine nucleotide exchange factor, to swap out its GDP for a GTP.
And once RAS is active,
it sets off this domino chain of phosphorylation. RAS activates RAF, which activates MEK, which activates a class of enzymes called MAPKs, or mitogen-activated protein kinases. The MAPKs then travel into the nucleus and change gene expression, often telling the cell to grow and divide.
To really study these pathways, genetics is key. And we need to know two types of mutations. First, a dominant negative mutation.
This one is insidious. The mutant receptor can still bind the ligand and dimerize with the normal receptor, but because it's broken, it prevents the normal one from working. It effectively poisons the whole system.
And the opposite of that is a constitutively active mutation,
where the receptor is just stuck in the on position, even with no ligand around. A classic human example is a mutation in the FGFR3 receptor that causes a form of dwarfism, a chondroplegia, because it's signaling constantly.
Let's tie this all together with insulin. Insulin binds an RTK, which phosphorylates an adaptor protein, IRS1.
IRS1 then recruits PI3 kinase, or PI3K. That enzyme generates another lipid second messenger, PIP3, from PIP2.
And PIP3 activates ACT, which is also known as protein kinase B.
And activated ACT is the crucial player for glucose uptake. It's what promotes the movement of GLUT4 glucose transporters up to the plasma membrane. It also promotes glycogen synthesis. It's a huge hub.
And just to wrap up, it's critical to remember these are not simple straight lines. These pathways interact
constantly. Cells use things like scaffolding complexes to physically assemble all the components of a single cascade together. This increases efficiency, and it also prevents crosstalk.
Which is when one pathway accidentally activates parts of another one.
Exactly. The main takeaway for you should be that cellular signaling is this vast interconnected network. The cell uses a surprisingly limited toolbox, kinases, phosphatases, G proteins, to pull off an infinite number of highly specific tasks.
We have covered a huge amount of critical vocabulary today, from the quick electrochemical gradients in neurons governed by the Hodgkin cycle and the Na plus K plus pump, to the cascading chemical messages of hormones, all dictated by Katie Finity, and the logic of GPCRs and RTKs.
Understanding these terms is really about understanding the molecular logic of life itself.
We mentioned earlier how memory formation relies on strengthening synapses, especially through calcium influx driven by receptors like NMDA. So if the storage and retrieval of our memories are literally physical changes tied to the strength of these chemical synapses, consider this provocative thought for your study break. If we can fully map the exact calcium dynamics needed for long-term potentiation, the process that strengthens a synapse could sophisticated tools like optogenetics eventually allow us to artificially insert or even rewrite memories by precisely controlling the timing and amount of that critical calcium influx. Something to think about.